All right, let's discuss factors that can affect skin color. So there are three categories of things that can affect our skin color. The first is being hereditary factors. So this is going to be your DNA. The second is environmental factors. These can be things that alter the expression of our genes. And then third are physiological factors. So this is just going to alter the appearance of our skin, but not actually affect the expression of genes or our DNA. So we discussed and I showed you where melanocytes are in the epidermis. So they're located in the stratum basale. And we all have the same number of melanocytes, but it is the amount of melanin that changes, that is produced, and that is under genetic control. So it is the amount of melanin that is produced that changes between individuals. And not only that, but melanin is packaged in granules. So the size and distribution of these granules can actually change. So there are mutations in the melanin gene, which individuals such as albinos have, that lack melanin. So they have melanocytes. They have the exact number of melanocytes that any other individual would have, but they are not producing melanin. So a true albino doesn't produce any melanin. So environmental factors are the second category of events that can affect the color of our skin. So in this situation, some people, go to the beach and sunbathe, or they go to tanning beds. And this increase of UV radiation will actually stimulate melanocytes to make more melanin. So again, there's not more melanocytes, there's just more melanin. And obviously, too, UV can cause DNA damage. So there has to be this very delicate balance, because we do need some exposure from UV to make vitamin D. So vitamin D is very necessary for our bone health, but we don't want too much of it in that we get cell mutation. So interestingly, there is somewhat of an epidemic of vitamin D deficiency, because kids and adults, teens are staying inside more and not getting sunlight. They're in front of their computers or doing indoor activities. And then there was this push to use so much sunscreen and big hats and long sleeves. And so individuals were not getting enough vitamin D for our bone health. And there became a collaboration between milk companies to start adding vitamin D. So that's why you see fortified vitamin D milk. Sorry, my screen just darkened a little bit. And then also cereals do the same thing. So environmental factors. And then the third is physiological. So this can change from moment to moment. So we have something called vasodilation and vasoconstriction of the blood vessels that are close to our skin. And this can happen when we're exercising. So the blood vessels will open up and cause our skin to turn pink. When we blush, if there's, an embarrassing moment and you feel embarrassed, your skin will blush. There's also the reverse. If you're frightened, the saying, you get white with fright. So you're having vasoconstriction, and your skin takes on more of a whiter appearance. And then there's also a dietary contribution to the color of skin. This can happen from carotene, so eating lots and lots of carrots. You'd probably have to eat bags and bags of carrots, but this could also affect your skin color. All right, so we are going to move on to the accessory organs of the integument or skin, and those accessory organs are hair and nails. We're not going to cover nails in this lecture because there's just too much to cover already. We will talk about some of the glands that are associated with hair as well. So this image over here is showing you hair. Hair is found throughout the body. We have several different types of hair. We have extremely fine hair called vellum hair. I'm not going to quiz you on that or test you on that. I'm just telling you about it. This is kind of what is referred to as peach fuzz, which is, on a baby or on an adult's face. Then we have traditional hair. That's called terminal hair on the top of your head, your arms, your back, your chest. And then we have particularly thick hair on our eyebrows and our eyelashes. So all of these types of hair have a very similar structure to them. Most of the hair is dead, similar to the stratum corneum. And there is also a partnership between the epithelium of hair and the connective tissue, which we'll go over. So this image shows hair cut in a longitudinal section. So you can see the epidermis right here. That's the epidermis. I'm just going to abbreviate it. Well, I'll just write it out. That's the epidermis. And then everything here is the dermis. Okay, sometimes hair will progress down into the hypodermis. So hair is divided into several regions. There is this hair bulb here. There is the hair root, which is outlined here. So it extends from about here to this dotted line. So what the difference between the root and then it turns into the shaft from here on out, the difference is that within the root, the hair is attached to the nearby structures, so it is kind of holding it in place. And then when it reaches the shaft region up here, it is more free floating in the channel, so that at that point, that's where it becomes the shaft. And then, so you can see it kind of detaches a little bit. There's a space here, and that is where the hair becomes the shaft. So those are the regions of the hair. Each individual hair is associated with a smooth muscle called the erector pili muscle. This is smooth muscle. Smooth muscle we do not have control over. All right, so each individual hair is associated with this muscle, and the function is not entirely effective anymore. So it was supposed to pull on your hair to make it stand up. So if all of your hair is standing up, you're supposed to look more frightening or larger. So animals do this, right? You see cats do this. Their hair will raise up so that they're really big and kind of scary. But we don't really have that response anymore. So the muscle isn't strong enough to make the hair on our head stand up. But you will see this muscle, and you should be responsible for identifying it both on histology and model. So that is what that muscle is right there. There's also glands that are associated with hair called sebaceous glands. I'll show you what they look like when we look at histology as well. And these are going to produce a substance called sebum. It's an oily substance that lubricates and moisturizes the hair. On a side note, it is also what can tend to cause acne as well. So the overproduction of sebum can oftentimes give us trouble with acne, and the the drug Accutane. If you've ever suffered from acne or know anyone who's suffered from acne and has been on Accutane, it's kind of a pretty harsh drug, but it does inhibit sebaceous glands from releasing sebum. So it does tend to help with acne pretty well, but it also causes really dry skin and hair. All right, so before I go over the histology, I want to take a closer look at the bulb. So this, so I also want to make sure, did I tell you, so that hair, most of the hair is dead, okay? So the only real living part is down here in the bulb. All right, so let's take a closer look at what's going on with this bulb. Okay, so what surrounds the hair? So what is seen here in kind of blue, purple is connective tissue, okay? So there is connective tissue. It is areolar connective tissue that surrounds the entire portion of the hair. It also pushes in here to form a papilla. All right, papilla means finger-like projection. Finger-like projection. I'm getting ready to spell that wrong. So it's a finger-like projection, the papilla. And that there are, as we know, in areolar connective tissue, there are blood vessels. And the reason why you get sort of this projection of the connective tissue is because this area that is closest to that papilla is the matrix. And this is where the dividing cells are located, kind of like in the stratum basale. And that is what is going to produce all of the cells to make the hair. So the stem cells and the melanocytes are going to reside in this matrix, all right? And again, this is what is going to produce the cells that will make the hair. So all of the structure from this kind of dark brown region, all of this is going to be the hair. Let's look at a histology section, and then I want to talk about a couple other layers that you're going to find in the hair. This looks like a complicated image, but a lot of this we've talked about already. So here are the layers of the epithelium, the epidermis, excuse me. We just mentioned the erector pili muscle. I'm going to go over the secretory glands at the very end. Here is the hypodermis containing adipose. Okay, you can see this nice structure. Here's the hair follicle here. And you can see this is the bulb portion. Here's another image showing you where the erector pili muscle is. And then hair is always going to be associated with sebaceous glands. I have several other images so that you can see, you'll see what to expect when you look at it under the microscope during lab. But I want to go over some of the layers that you're going to find when you look at a hair follicle in cross section. All right, so there are three essential layers that make up the follicle. So we have the cortex, the medulla, and the cuticle. So in the innermost region is called the medulla. This is the core of the hair. The cells here are making a flexible, soft type of keratin. The cortex contains cells that are making a harder, less flexible keratin. And this is what's going to give hair its stiffness. So these two layers kind of define the type of hair that you have. So if you have thick hair, you might have a bigger cortex. If you have thinner hair, you may have more of the medulla layer compared to the cortex. So that is what is defining kind of the thickness or thinness of your individual hair. And then the cuticle is the outside layer. This is a very tough, hard keratin. It's very thin though, it's about 1 cell layer thick, and it is a protective layer to protect the cells underneath. All right, so here are the layers if you are to look at them under histology. Let me make this a little bit smaller so you can see. So when you're looking at it under the microscope and you're able to see a cross section, so you're going to see both longitudinal sections and cross sections, right? Because we can't all line the hair up in one particular order. It just depends on how the cut is made. So lots of times you'll see both. So when, I think this image is probably a little bit better, it shows all of the layers. You can see the medulla here being in the middle, and then the cortex layer, and then the cuticle surrounds it. And then you can see kind of the connective tissue that is around it. This is another really nice image showing you this is now a longitudinal section of hair. This hair is probably on some kind of diagonal path, so that's why you see it like that, but you can see the bulb, you can see the hair here, and then the shaft of it. This is a close-up of the bulb. You can make out that papilla here. And then closest to that are those living, dividing cells of the matrix that have the melanocytes and the stem cells that are going to go on to produce the rest of the hair. Here's a nice image of sebaceous glands. This is a sebaceous gland right here. They look really remarkable under the microscope. You should be able to see them without any problem. I've gone over this already. It softens and lubricates the hair. It produces a substance called sebum, which is an oily substance. And these sebaceous glands are anywhere where there is hair. Again, the only place you do not have hair is are located on your palms of your hand and the soles of your feet. This is another nice cross-section image of hair, which is showing you the follicle and the hair shaft, and you can see the sebaceous gland located right next to it. This image highlights the erector pili muscle. Sometimes they're hard to make out when you're looking at these images under the microscope. I'm not going to kid you, but it is worth looking at. All right, so the last kind of topic I'm going to talk about are the glands that are associated with skin, so sweat glands, and we'll wrap up the lecture with that topic. I want to discuss with you a couple of the different types of glands that you will find in skin. So we touched on a couple of these already. And so let me point out the important features that I want you to know. So there are two types of categories, or two categories. The first is sebaceous glands, which I touched on concerning hair, but they are also associated with skin. And then there are sweat glands. So let's work on this side first, and then we'll move to the second. And then I have a summary slide after this to kind of summarize everything I just went over. So we talked about what sebaceous glands do. I was mostly referring to the sebaceous glands that are associated with hair follicles. They secrete that oily lipid called sebum that coats the hair shaft and the epidermis, and it provides lubrication to the hair. You can also find the sebaceous follicles on skin to kind of help lubricate the top surface, that stratum corneum of our skin. So that is why, if you work in an environment where you have to wash your hands a lot, like in the hospital, they can crack and bleed because you're washing away the oils that are trying to keep this stratum corneum healthy. So those are the sebaceous glands, again, associated with hair as well as skin. And then we have sweat glands. This is more of a watery substance that is being secreted. And there are two types. I'm going to change this word here because I use a different word. So there's what's called apocrine sweat glands and eccrine sweat glands. And the apocrine sweat glands are limited in their distribution. They're mostly found in the axillary region, the armpit region, the groin, and a little bit around the nipples. They're producing more of a viscous solution rather than like a really thin, watery solution. And this is also the source of human pheromones. They also have like a antibacterial effect to it, and because they are this secretion contains more than just water. So there are some salts and there's some substances like proteins. They can have like some growth of bacteria to it, which can lead to body odor. So that is what is apocrine sweat glands. They are associated with hair too. We're going to skip this region or that portion, those types of glands. And then the eccrine glands are like your truer sweat glands. They are really abundant. They're widespread. This is like your kind of true sweat when you're working out that is produced along your body. They're, again, widespread. This is the saltier secretion, and this is important in thermoregulation. Right there. So I've summarized these types of glands here. Again, so the sebaceous glands we went over, they're distributed between all over the body and associated with hair follicles as well as the surface of the skin. And then the two types of sweat glands, this is more like your true sweat glands, is the eccrine sweat glands. Sorry, my dog is moving around, my stove is going off. It's not easy to record these lectures and have sort of a quiet environment. Okay, so the true sweat is produced by the eccrine glands, and then the apocrine glands is more associated with the axillary, anal, genital areas. This is producing the sweat that has some oils, some proteins, lipids, and bacteria can sometimes grow here too, and this is producing more of like the body odor. All right, and then the last topic is skin cancer. I think this is an important topic to go over before we end this lecture series on skin. So there are three types of skin cancer. There's what's called basal cell carcinoma, squamous cell carcinoma, and then melanoma. Basal cell carcinoma is really the most predominant one. That's the one that makes up about 80 to 90% of all cancers, but it is highly treatable. I'll show you images on the next slide. Squamous cell carcinoma, this arises from the keratinocytes, from the stratum spinosum that we talked about kind of extensively. This covers about 10 to 20% of skin cancers. It is a little bit more aggressive, and it's something that should be monitored, but it is highly treatable if it is caught in time. And then melanoma, this is deadly. This is cancer of the melanocytes. There are a lot of skin cancer deaths related to melanoma if it is not detected early. So let me show you kind of what this looks like on a picture format. So, here are layers of skin that we've talked about throughout this lecture. Here are the two treatable ones. So we have this basal cell carcinoma and squamous cell. You can see that they kind of dip through the epidermis and they kind of begin to get into the dermis layer. If these are caught early, they can be removed. The underlying fear between like what makes this so deadly is that when these kind of cancerous lesions kind of enter into like the connective tissue region where there is a blood supply, or whether there's a lymphatic system where there's vessels located nearby. When these cancer cells can then enter into those vessels or vascular system, they can then travel throughout the body. So that is kind of the worry, and why, the depth of how far these, typically they're moles, migrate into the skin is of the biggest concern. So that is why early detection is really important, so that they don't start migrating and progressing deeper into the skin. So there's these rules. It's called the ABCDE rule to kind of monitor. And for whatever reason, these images turned blue. They should be fine on your PDF version that I will attach, but I don't know why they kind of showed up blue here. But the important thing is just reading through this to note that, so this happens with moles. So what you're supposed to do is kind of follow these rules, and you follow moles over time, because some moles will never develop into cancerous, like melanoma, while other moles will. So you want to kind of follow the outline of the mole and make sure that the symmetry of them doesn't change. So once they start to begin to look a little asymmetrical, that is when you should have those looked at by a dermatologist. The border of the mole is also critical. So if the border starts to change, that's a sign. When the color of the mole, if it darkens, that's another sign. And then obviously the diameter, the bigger it gets, the more worrisome it is. And then E stands for evolution, so kind of monitoring it over time, over months or years. If the mole begins to change, that is a concern as well. And then finally, talking about burns, just kind of bringing some relevance, some clinical relevance to these lectures, I think is always important. So this outlines the severity of burns. As you know, they're classified into first degree, second degree, and third degree burns. And then there's a nice picture here that shows you what layers of the skin is being damaged by these burns. So first degree burns really only damage the epidermis. This is like a sunburn. It's not incredibly painful. It's a little bit uncomfortable. Secondary burns are quite painful. So this burn has burned through the epidermis and deep into the dermis. And what happens is you do get a lot of blisters and bleeding. This is pretty painful because you can see in yellow, when I don't remember, I feel like my hypodermis slide wasn't great, but there are blood vessels in the hypodermis, and there are nerves, and you can see how these nerves kind of like come up into the dermal region. So you will have the sensation that the nerves will detect this pain. It will be processed in your brain, and you will realize that this is a very painful type of burn. Third degree burns are interesting because all of the layers of skin have been damaged, And since you have damaged the hypodermis region, you have pretty much killed all of these nerves. Like, you've damaged the nerves so much that they are not sending signals to the brain anymore. So these tend to be, although they are very dangerous and can be very problematic, they are less likely to be painful because the nerve endings have been destroyed. So they are not signaling back to the brain. However, there really is not much you can do about repairing this skin except to get a skin graft, which, you can take skin from other parts of the body and kind of attach it back onto the region that was burned. So these kind of burns, even though they're painful, they will heal on their own. Third-degree burns, you are left with very few options, and then one of those being skin grafting. So there are labs that are studying skin and trying to grow skin in vitro in a culture dish so that you don't have to take it from your own body. But that is kind of where there are new studies coming out trying to develop skin within the lab. So that is a summary of burns and how the skin is affected and what layers are affected. Please make sure that you know which layers are affected and what happens with each of these. All right, I think that concludes the lecture on skin, and I hope you enjoy it, and we will see you in lab.